to isotopic analysis, to bioarchaeological materials. It'll be a sort of general overview, because there's parts of these kind of things that they could be whole lectures by themselves. So it's more to provide an overview of what isotopic analysis are, and then how we can apply them to archaeological material, and particularly archaeological research questions in Turkey and the Near East. So isotopic analysis are not just something that we use in archaeology, they can be applied to a multitude of things. So in archaeology, for example, we can look at them, use them to reconstruct paleodiets, looking at subsistence and agricultural practices, migration, mobility, residency patterns, the provenancing of archaeological materials, trade and exchange networks, animal management and herding practices, and also the movement or trade and exchange of animals and plants as well. They can also be applied to ecology, so aquatic and terrestrial ecosystems, food webs, movement and migration of animal species. Also, we can use them to reconstruct paleoclimates and paleoenvironments. They can be applied to geology, hydrology, also in modern ways. They're applied to like foodstuff quality controls and checking the source and origin checks of foodstuffs. They can also apply to forensics as well. Things like not just sort of identifying things to do with victims of homicide, but also drug control and these kind of things. So they have a wide range of applications, and they weren't initially kind of discovered and applied to archaeology, but archaeologists realized the utility of them and how useful they could be for answering research questions. So you'll hear this word a lot today in my talk about bioarchaeology, and obviously the titles applying isotopic analysis to bioarchaeological materials. In the American term, bioarchaeology just means human bones and things around human biology and what we can learn from the human skeleton and within its context, in archaeological context. However, the European, the UK term, which is the one that I use, is bioarchaeology, refers to all living things basically within an archaeological context. As you can see on the slide, it's the study of bones and other biological materials found in archaeological remains in order to provide information about human or the environment in the past. So basically this means humans, the holy trinity of human, plants and animals. So if we then focus on sort of archaeology, bioarchaeology, so as I've said, when it's bioarchaeological materials is humans, animals, plants. You can also apply them to inorganic materials, but this talk won't cover any of those things today. So they use them for provenancing of things like pottery, metals, marble, these kind of stuff. But we're not going to cover anything of that today. So in humans, they can be, like I said, they can be used to do paleodietary reconstruction. And this means they can provide indications of the source and relative amounts of consumed food resources. And then from this, you can then start to delve into deeper questions about socioeconomic and paleodemographic dynamics when you look at population level and also regional levels. So are there differences between different parts of the society, different levels of the population? Are they different between regions, time periods, diachronically, these kind of things? So when you apply them to humans and animals particularly as well, you can look at subsistence strategies, so how humans manage animals, human and animal interactions, so animal husbandry, animal management, and then also in plant remains, they can provide indications about the local environment and climate. So this is partly where there's an overlap between archaeology and also things like climate and environmental reconstruction. So we can use isotopic analysis applied to the plant materials for that. And then also as well, we can look at management of plant remains or botanical remains. So how people made choices about their crops, their field location, what they were growing, where they were growing it, how they were growing it, these kind of things. And then obviously, as I mentioned, we can also apply isotopes to bioarchaeological materials to examine the movement and mobility of them. So we start at the very beginning, I guess. And so what is an isotope? So atoms consist of protons and electrons. And so the electrons is the Z number, as you can see on the picture on the right side of the slide here. So the number of protons is the atomic number, which is Z. and the number of neutrons is n, and the Z plus n gives you the mass number. The atoms with the same Z number but a different n number, and therefore obviously different A number, are isotopes. And these isotopes have more or less identical chemical properties, but they have different masses. And most elements, or many of the elements, have at least 2 or more naturally occurring isotopes. So for example, if we look at carbon, the element of carbon, so you see here like the atomic number is 6, but the mass number is 13 because that's to do with a different number of neutrons. So you have things like carbon 13, carbon 12, and obviously carbon 14, which I'm sure most people are familiar with because that's what's used for radiocarbon dating. So stable isotopes are measured by mass spectrometry. That's the best way to do it. And so this slide's going to give you a bit of background. So you may see these things in publications or mentioned, and it's just to sort of help understand a little bit. So after the mass spectrometry, stable isotope ratios are given in parts per thousand, more commonly known as per mil, and that's that sign there. It's like a little percentage sign with an extra little 0 at the bottom, and this is relative to a 0. So for example, for delta carbon 13, this is the standard that's used is the PD belemnite standard, PDB, and for delta nitrogen 15, this is the ambient air reservoir air. So here for the carbons, the carbon 13 abundance ratio is the ratio between carbon 13 and carbon 12, and then the equation below is the standard equation for calculating your delta value. So your delta carbon 13 in parts per mil is the ratio of the sample. So that's what you're analyzing, divided by the ratio of the standard. So obviously in carbon, as I mentioned, this is the PDB, minus 1, and then times 1,000, that's what gives it per mil. So the most common isotopic ratios that are used in archaeology, applied to bioarchaeological materials, are carbon, nitrogen, sulfur, oxygen, and strontium. However, more recently, there's been the examination development into the utility and application of what are more commonly known as novel isotopes. So these include things like calcium, magnesium, copper, iron, lead, and hydrogen. And as research into these kind of things develops and continues, it's likely that the sort of standard repertoire of carbon, nitrogen, sulfur, oxygen, and strontium will be expanded to include these other isotopic ratios. For example, hydrogen is becoming more common in recent years to be applied to questions. However, in this talk, I'm just going to deal with the standard sort of set that are commonly used at the moment. So if we first go into carbon, so this has 2 stable isotopes, I've already mentioned, carbon 12 and carbon 13. And as I've also mentioned as well, so the isotope ratios are expressed as delta carbon 13. And carbon is very useful because you can use it to distinguish between C3 and C4 plants because they have almost unique distinct values. So the average of C3 plants is about minus 26 per mil. And these are things like your common plants of trees, crops like wheat, barley, for example. And then you have C4 plants, and they have a sort of average value around about minus 12 per mil. And C4 plants, most people will be more familiar with what called the New World crops, so things like maize. But in the Old World, so if you think about Turkey and the Near East, C4 plants would be things like sorghum, millets, and there's also wild C4 grasses as well. Most delta carbon 13 values in biological systems are negative, and as well as being able to distinguish between sort of C3 and C4 input into animal plant diet, they give the source of the carbon. So I say mainly whether it's C3 or C4. Also things to do with the plant stuff. I won't go into too much detail about plants because I know Tom in the next talk is going to go into more detail about that stuff. But also the carbon is useful for the humidity levels during the growth of plants. So particularly it's applied to crops and trees, and then we can see, this is what I was talking about before, about how crops are managed, where they're grown, how they're grown, stuff. As I said, I'm not going to go into too much detail about this because Tom is going to cover that in the next talk. So nitrogen has 2 stable isotopes, nitrogen 14 and nitrogen 15. Again, like carbon is expressed in the delta nitrogen, this is delta nitrogen 15 in this case. And nitrogen isotopes reflect the trophic level and the contribution and source of dietary protein. The marine diets usually exhibit enriched delta nitrogen 15 signals, and I'll give some case studies later on to demonstrate this. Most delta nitrogen 15 values in biological systems are positive, unlike carbon. So this is just to sort of show you, if I can get the, here we go. So this is a terrestrial, on the left is a terrestrial food web, an example, just a hypothetical one, and on the right is a marine one. So you have here your primary producers, plants, and basically as you go up the trophic levels in a food web, there's about a +3 per mil difference between diet and consumer. So if we start here with the plants and then the herbivore, this little rabbit, they would have a +3 value compared to the plants, and then into the omnivores and carnivores, another +3. So you see plants will start here at about a 2 per mil in this example, and then the rabbit would be about 5, and then up to like 8 for the carnivores and stuff. And then the marine, it's the same principle. As you go up the trophic levels, you get this +3 per mil increase. And the reason that the delta nitrogen 15 values are higher in marine ecosystems is because there's more trophic steps, so there's more chances for this +3 to take effect. So by the time you get to sort of a top predator, they've already gone through more steps than they would in a terrestrial environment. So the next one is sulfur. I'm going to give a little overview about this is the ratio. So delta sulfur 34 is the ratio of sulfur 32 to sulfur 34, and the standard is used. This used to is compared relative to is the Canyon Diablo troilite. Delta sulfur 34S can be used for diet provenance, so where the food came from. And then this means it can distinguish between, it's also quite useful for distinguishing between freshwater, marine, and terrestrial. Whereas carbon and nitrogen, you can distinguish between different levels of the diet or different sources of the food. It's difficult sometimes to see between terrestrial and freshwater. Whereas terrestrial and marine are quite distinct with carbon and nitrogen. Terrestrial and freshwater are more difficult, and delta sulfur 34S, sorry, can help with this. This means it can also be not only just used for diet provenance, but also you can then use this to apply it to mobility questions and residency. Basically, you see the difference between coastal and inland dwellers. So as you can see here, you have sort of it's about a plus 20.3 is in marine is the around +20 is sea spray, and so for seawater, and then obviously the further inland you go, this changes. It's very variable. It can be very variable, and it's based on the isotopic composition of geological bedrock. So this just to give a little quick example. So as you can see here, the example between marine and freshwater, we see there's two distinct values more or less sort of determined. And then this example here from an article by Nalick in 2015, you can show that so this is often this is delta sulfur 34S, and this is delta carbon 13. And when you sort of plot them together, you can see the terrestrials group, and then the freshwaters group, and the marine groups here, and then this is the sea spray. So the closer you get to sort of 20 per mil for sulfur, the more likely you are to be closer to a marine environment or come from a marine environment. And obviously the reverse is applicable, so that the sort of the further away from 20 per mil you get, the more likely it is to be terrestrial. So now we move on to oxygen, and this is looking at the ratio of stable isotopes oxygen 16 and oxygen 18, and it's compared to the Vienna Standard Mean Ocean Water, VSMOW. Again, expressed in the delta as delta oxygen 18. And oxygen, we use it because it reflects the isotopic composition of ingested water. So whether it's an animal or human, it gives you the oxygen signature, the isotopic signature of the water that's been ingested. It's very highly variable, but when you sort of tease it out and understand it, you can be used to reconstruct seasonality, geographical origin, patterns of migration, and also you can use it for paleoclimate reconstruction. So you can see here what happens is when it evaporates, the lighter oxygen-16 isotope is more easily evaporates, and then conversely, obviously, with rainwater, oxygen-18 is preferentially removed by precipitation. You can see here some sort of like a rough schematic thing of the different kind of values to do with rivers, ice, the sea, clouds and rain, etc. So by understanding these kind of basic values, and then when you apply them to archaeological remains, you can examine things like I said, like mobility and origin. Again, Jana Eger, who will be talking in a couple of talks, she's going to deal with oxygen a little bit more and applying how it can be applied. So I won't go into too much detail about this either. And the same for strontium. I know that Jana is going to be talking about strontium and applied to animal remains. So I'm just going to give a basic overview of what strontium isotopic ratios we use. So it's the isotopic ratio of strontium 87 to strontium 86. 87 is radiogenic, whereas 86 is stable. So because it's not a stable isotope ratio, it's not given in a delta value, unlike the other ones that we've looked at so far. Strontium comes from the geological bedrock, like sulfur. So it's based on the isotopic composition of the geological bedrock, and it's incorporated into bone apatite and dental enamel as a substitute for calcium. Ratios in dental enamel are most commonly examined, and I'll explain why in a bit. But like I mentioned, you can also look at it in bone apatite. So that's the inorganic mineral part of bone, which I'll also explain a little bit as well. Strontium is almost exclusively used for mobility studies. So what it does is it gives you the geochemical origin of what you're sampling. So if the bedrock in one area is has a particular strontium isotope ratio signature, but then you find that person in an area with a different signature, then you know that person is not local to where you found them and sampled them from, but came from somewhere else. And if you have a good strontium isoscape mapping, then you can sometimes even identify where this person came from. So we now move on to sort of sampling and which parts of animals and humans you would examine isotopic ratios in. So as I sort of briefly alluded to, you can measure isotopic ratios in the inorganic and inorganic tissues of bones and teeth. So it's organic part of this in bones or dentin of teeth is collagen, and then in bones this would be the apatite. So generally carbon-13, delta nitrogen-15, delta carbon-13, and delta sulfur-34S are from bone collagen. This can be bulk bone collagen. So this is just the general, you extract collagen from the bone and then you analyze it. You can look at, for dental enamel and bone apatite, this is usually, you can look at delta carbon-13 and delta oxygen-18 and strontium 87, 86. You can also, this is another recent development, but scientists have developed ways of no longer looking at just bulk bone collagen, but also looking at individual compound-specific amino acids from within that collagen, and applying delta carbon-13 and delta nitrogen-15 analysis to amino acids. You can also apply isotopic analysis to other biomaterials, things like dental calculus, which you find on the teeth. So to go into a little bit more detail about this, so we mentioned collagen and apatite, so organic versus inorganic parts of the bone. And as I also mentioned, collagen is made of amino acids, and collagen reflects the protein source of the diet. So obviously, as collagen and amino acids are a protein, so it's the sources of protein in the diet that you get from collagen. Apatite reflects the overall diet, so this includes as well as proteins, lipids, and also carbohydrates. So obviously, everyone would say then, oh, apatite's the best thing to analyze. Then we should be doing apatite because that gives you an overall reflection of the diet, of consumed food resources. However, there is a problem with apatite in that it is more susceptible to post-mortem diagenesis than collagen. So the risk, if the isotopic signature that you are getting from apatite in a bone isn't actually related to what the individual, that whether it's animal or human, is consumed, but actually to the burial environment, to the soil, water, these kind of things around the bones. So collagen is generally used for things like paleo analysis, because it's less susceptible to these kind of diagenesis things. But we do have to be aware that bones are not static. Bones are not like concrete or whatever. They're constantly modeling and remodeling, and this results in bone turnover, the turnover of bone tissues, and therefore obviously the turnover of collagen, and then obviously as well this gives you a turnover of isotopic signatures. So isotopes, isotopic values in bones, from collagen particularly as well, they typically represent the lifetime average. More accurately, this is the last 10 years of life depending on the bone. So if you take it from a bigger bone, something like the femur, so that's the large leg bone here, this will give you, yeah, it's about an average of about 10 years. If you take it from a rib, this has a faster turnover rate, so you're probably getting like an average of 5 to 7 years. So actually one of the things you can do is you can sample from the same individual. You can sample from a long bone, like a femur, and also a rib, and compare them and see if, you know, in that sort of slight difference in turnover rates, where there's any changes to diets in that sort of, you know, 3 to 5 years difference. Enamel, unlike bones, are not remodeled in life. So once you develop your enamel as a child, that's it. So this is why I was mentioning with strontium, it gives you this origin. So it's where someone grew up, it's where someone was born and spent the first, depending on which tooth you analyze, the first sort of 3 to 5 years, maybe a little bit more, a bit less, like I say, depending on the tooth. But that's the signature. The isotopic signature you'll get is from that phase of growth, because after that it no longer remodels. So as mentioned, the plants. So these are the prime producers in trophic systems, and as I've talked about, we can look at paleoclimate and paleoenvironment with plants, and then also anthropogenic factors such as agricultural practices. So this includes things like irrigation, manuring, field management, and also trade and exchange networks and regional interactions. With plants, most common isotopic ratios applied are those of delta carbon 13 and delta nitrogen 15, but also there has been some applications of strontium to archaeobotanical remains to examine movement of plants. So once you've one of the things, and these things were discussed yesterday and some earlier talks, it's important when you're doing these kind of like environmental and archaeological science, applying these techniques, the first thing to think of is your research question. What are you looking to find? What do you want to find? What are you trying to answer? What are you trying to investigate? So therefore you would choose an appropriate isotopic ratio. So you wouldn't apply strontium or oxygen necessarily, wouldn't be your first choice for examining paleo diet in humans or animals. So do you then have to work out which materials you want? So do you want bone, collagen, apatite, or dental enamel, depending on your research questions, and then also your sampling strategies. And then you have to then, depending on what you've chosen, which ice and which material you're examining, you then have to treat them. You can't just simply take a bone or a tooth and run them through a machine and expect to get results. So, but this does depend on the isotope being applied and also the material being studied. So for human and faunal, skeletal and dental remains, it's obviously the first step would be to remove a sample. You don't need the whole bone or the whole tooth, you need part of it. Usually about 5 grams is enough, I mean more than enough, but you don't have to remove more than that. Because isotopic analysis is a destructive process. So once you've sampled that dental or bone tissue, that sample will then be destroyed. So the less material that you can take from a skeleton, whether it be human or animal, is better, because like I say, it's destructive. As I also mentioned, for bones, you normally take fragments of long bones. So this would be slow turnover bones. So things like the leg, arm bones, and then fast turnover bones as well. So these would be ribs or phalanges, that's your finger or toe bones. And for dental stuff, it's usually molars. And if you're sampling bone, it's best to take cortical bone. So that if you think of a bone being like a circle like this, the cortical bone is the outside part. And you want to take from a nice thick part to ensure, especially for collagen, because there's more chance of having good collagen quality preservation in there, and a good amount of collagen strands in there. It's also good to avoid pathological or modified bone. So you want to avoid sampling burnt bone, bone that has been excessively weathered, whether by the sun or water. And then again, depending on what isotope you're examining and which material, the extraction and treatment protocols differ. But generally, this includes some sort of cleaning to get rid of contaminants. In the case of collagen, there'll be a demineralization stage, and then refinement treatments as well, so that you kind of make sure that the results you're getting, so the values that you get, are actually, isotopic values of the plant, human, animal, rather than the surrounding archaeological context, burial environment, for example. So if we take collagen, for example, collagen from bones, this can be human or animal, just to sort of give a very rough and brief overview of the process. So the first step is that you would sample, obviously, the tissue you want, so the bone tissue. You then make sure you clean it of external contaminants, so let's say things like mud, dirt, calcite, these kind of things. It then goes into a demineralization phase, which is this first picture here. So then you put the bones in hydrochloric acid to get rid of the inorganic part and demineralize them so they go soft. So you have like a hard bone, and after they've been in the acid for a few days, they then go soft, a little bit rubbery. And then later you have to change the acids, clean them, filter them. They then go through a heating block to gelatinize the solution, and then into a freeze dryer, which is this process here. The freeze dryer extracts all the liquid and moisture, and then hopefully, if everything's gone right and you have good quality bones, you'll be left with collagen here. So it looks a bit like candy floss if it's good. It's kind of white or creamy and fluffy, and then this is what you then put through a mass spectrometer to analyze it. And then here, if you can see here, this is like a little tin capsule, and you then take, so this is a whole, this is how much collagen came out of one bone, and you don't need a little bit of this, and then you put 0.5 milligrams, something like this, and you put it into a tin capsule, and then that's what you'd apply to the input into the mass spectrometer. So as well as sampling the population of individuals that you want to analyze, it's important in isotopic studies to do extra sampling as well. So it's important to obtain your baselines or your isoscapes for a regional site. So this can be taking isotopic data from plants, animals. If you're looking at human diet, for example, you want to know, obviously, because there's variations in isotopic signatures for plants and animals. If you don't know what these signals are, then you might not be able to fully interpret the isotopic signals of the humans and work out their dietary inputs. And if you're doing mobility and residency studies, then you need to figure out what is the isotopic signature of your local area, of the site, of the local region, so that you can then identify non-local people, people who are more mobile than others, for example, as well. And this means that isotopic analysis becomes quite a collaborative effort. So you have to, if you're not for example, a specialist in human remains or animal remains, you then have to collaborate with the human osteoarchaeologists, the physical anthropologists, and also the archaeozoologists, so they can not only identify the species of the animal, or which part of the bone, or the age and sex of an individual, to help with your interpretations as well. And the same thing with, obviously, with plants. Unless you're an archaeobotanist, you need to collaborate with the archaeobotanists, so they can tell you what kind of plant this is. Are there any distinguishing features about your seed samples, for example, or your charcoal samples, or whatever it is you're sampling? And obviously then as well, you need to collaborate with the archaeologists, whether it be the excavators, the project directors, this kind of stuff. So you can identify and gain more knowledge about the context of where your samples come from. If you're simply taking samples in isolation, then yes, you're going to get some nice data, and you can probably tell some basic things about it, but it only really becomes very useful once you kind of apply all these different facets. And so isotopic ratio analysis, sorry, as well as being sort of collaborative, it's holistic. It's not just one thing. It's one thing within many things. So after you've done all your sampling and you've got your collagen or your bone or enamel powder and it's all ready to go, you then go input it into a mass spectrometer. I'm going to go into too much detail again. Like I said at the beginning, these are the kind of things that you could talk, for a whole lecture about. But basically, mass spectrometry, it separates charged atoms and molecules on the basis of their masses in a magnetic field. And then this gives you the relative isotopic abundances, and the isotopic ratios and isotopic composition can then be calculated. So this here is an example of this is an isotope ratio mass spectrometer. So shortened, it's IRMS, and this has an elemental analyzer, so EA. This is commonly what is used for obtaining carbon, nitrogen, sulfur isotope values. So once you've done all these processes, you've figured out your research questions, you've sampled your humans, animals, plants, whatever. You've done your baseline sampling, you've prepared all your materials, you then get the data and the results. And then, so I'm now going to go into a little bit, how would you interpret these results once you get your data through? It usually comes in sort of something like the form of an Excel file of just a load of numbers, but then you need to sort of visualize it, help with your interpretations. So I'm going to use the example of carbon and nitrogen. So when you have carbon and nitrogen values, and more often than not, especially if you're looking at humans, animals, they're given together. You wouldn't just do one or the other necessarily. So carbon would be on the x-axis on the bottom here, then nitrogen would be on the y-axis. And then here you have your C3 plants. This is -26 we talked about. And as you see, C4 plants here, around about -12. And then obviously going through the food chains, increasing trophic levels. So this is a terrestrial food chain, as we saw before. This is a marine one. So generally, as you go more positive in the carbon, this can either be an indication of increasing C4 input into the diet or a marine input. And obviously, as you go up the trophic level, you're more likely to consume more dietary protein. So we give here, there is, as we mentioned before, there is an enrichment in nitrogen. So you have this +3 as you go up the trophic levels. There's also a very slight enrichment in carbon. So generally between diets, so whether it's plants or animals, and the consumers, you'll see this slightly sort of diagonal to the right movement. So if your animals here, for example, your humans should be around here. So you have a +3 and then a +1 to +2 per mil increase in carbon as well. Generally, if your humans were consuming a terrestrial C3 based mixed diet, so that's a diet composed mostly of terrestrial C3 plants and also animal protein, terrestrial animal protein, their value should be somewhere in this kind of range. This is what you'd expect to see. So something between about -18, -19 to about -23, -24 for carbon, and somewhere between about 5 and 10 per mil for nitrogen. Conversely, if you had humans with mostly marine diet, you'd expect them to see them up here somewhere. So they'd have this sort of more positive carbon value, and also this much higher, more positive nitrogen value as well. Usually anything above about 11 or 12 per mil can, in conjunction with a more positive carbon value, can be a possible indication of a marine input in the diet, or a mostly marine diet. And then obviously C4 as well. So studies have shown that there's generally a kind of cutoff of about -18. So the closer you are to -18, the more likely it is to be a C4 input. And the further you go, the more positive in carbon you become after -18, the greater the input of C4 plants in your diet. So for example, here on this chart, this is a human eating only C4 plants, and they expect to be here somewhere. So somewhere around -12 with the plants, maybe even -10, but with a low trophic level, so they're eating just C4 plants. So now look at some case studies of applying this to actual archaeological material. So this is some of my data for my PhD, and also an article I published. And this is early Bronze Age populations from Anatolia. So we have Iketepe, Tichusuk, Bakutepe, and Badamaji. And this is carbon nitrogen, looking at diet. And as you can see, they're in this sort of range between sort of -19 and -21, and between about 5 and 6 to about 10 per mil. So we can tell from this that these individuals were consuming a terrestrial C3-based mixed diet. And you can also apply carbon nitrogen to the weaning process. So this is when infants start to have the input of other dietary resources other than breast milk into their diets. And it's important for understanding social demographic dynamics, so things to do with birth spacing, fertility, these kind of stuff. So the nitrogen, so basically it works on the concept that the infants who are breastfeeding, who are suckling, are basically a trophic level above their mothers. Here are the adults, and then this purple circle is females in this site. So they should have, if they're breastfeeding, if they're solely breastfeeding with no other dietary inputs, they should have a +3 per mil difference between the mother and the child. So as you can see here, these infants here are still breastfeeding, and then as their values become closer to the range of the adults, we see you're more likely to have an input of other dietary resources into their diets, and when they fall within this range, this means they're consuming the same food resources as the adults. So then you can then see when does the weaning process begin. So at this site, it's probably about 1 year old. So this is when we start to see this fall in nitrogen. Carbon is the same principle, and actually studies have shown that carbon is more probably has a quicker reaction to changes in the diet. So it's actually even more useful than nitrogen. But the same thing, you'd have a sort of this +1 to +2 per mil difference. So these infants here are still only solely breastfeeding, and then again by about 1 year old, they are starting to fall in line with the adults, which means they're starting to eat similar foods with a similar isotopic signature, at least, to the adults. Sulfur, so carbon and sulfur again, and this is used to look at like dietary provenance, and it can give you implications about mobility, which I mentioned. So, for example, in this here we have the humans from Bakutepe, and these are the animals. This is their means, and they have slightly different variations in carbon. So they have different carbon sources in their diet. However, their sulfur values are very similar. So even though the animals are maybe consuming plants with a different carbon source, so this maybe would imply foddering or different grazing patterns, they're from the same area. So they're within the same area. So the animals are quite close to where the humans are. Whereas at Badamaji, it's the opposite. So we have, again, this kind of variation in carbon. So there's different carbon sources, but their sulfur is also different. So some of these animals came from a different region with a different sulfur signal, particularly some sheep and goat and a cow. These are their kind of values roughly plotted on this chart, especially this cow and the sheep and goat had very different sulfur values to the humans. So these animals for sure came from a different area, or at least to be more precise to say an area with a different sulfur isotopic signature. And finally, just to leave you with isotopic research in bioarchaeology in Anatolia. So these are the number of publications that have dealt with or include isotopic analyses of bioarchaeological materials. And as you can see, the first paper was in 2003. This was a paper by Richard et al. About humans and animals from Çatalhöyük. And generally, there's been a very positive increase throughout the years as more Turkish research projects and studies and researchers are applying isotopic analysis to their work. And then a little bit further to break this down. So what are the kind of research questions that people are applying isotope analysis to in Turkey, in Anatolian archaeology? Mostly the Neolithic and the Chalcolithic, and this is kind of to be expected, obviously, with parts of Anatolia and even Anatolia as a whole being involved with sort of the the domestication of plants and animals, the Neolithic process. So there's obviously an area of big interest to people is how did diets change maybe, and then what can isotopes tell us about this dietary change between sort of hunter-gatherers and sedentary agriculturists. And also, so what kind of things are people looking at? Again, mostly human dietary habits. And again, this is sort of linked in with this sort of Neolithic interest in the Neolithic. How is human dietary habits changing? But also as well, obviously, things like animal management practices. So how does the management of animals change, or human-animal interactions, and also animal mobility and provenance. So pastoralism, things like this, examined throughout different epochs of history and prehistory in Anatolia. And that's all from me. So I say thank you very much. And if you have any questions, I'd be very happy to take them. Thank you.